So anyway, dance is Saturday. It's at the clubhouse, the front here. I got my big sound system over here out and ready. We have yummy snacks. Lots of fun. We'll play whatever music you want. Oh, something there. It's a piece of Ozzy's. It is. I think he's been chewing on. It should be a lot of fun. You should come if you can. There. And buy the tickets on our website. They're less expensive to buy now. If you buy them at the door, they're more. So we buy them now, and then you. Oh shoot! He forgot his phone. I'm gonna go hand Jason his phone because he just left it down here. Be right back. So wait, I'm going to be back. Beautiful. Yes. So quarter of this glasses. Yes. That's me. Because we're really present. Yeah. Yeah, there you go. So what'd you do over the weekend? Rehearsal? Anything fun? Nothing? You what? You learned to dance for what song? Okay, good. What kind of style would you say it is? Is it ballet, contemporary, modern, tap? Just kind of a mix, or the choreography? How would you describe it? No? Kind of a mix of everything? Typical show tune stuff? We have a friend who's a Broadway choreographer, and it's always fun to see what he comes up with for different shows. He's really good. He's been in a few shows too, but he does a lot of choreography, and he's a really cool guy. It's fun to see what he does. He does a lot of like for this in the summer and stuff, he does a lot of regional theater projects too. He'll go and he'll be like the director and the choreographer for whatever city's summer music theater series, and he'll go do the whole series and travels around and does fun stuff. He's a really cool guy. Really kind of neat. Yeah. He's one of my husband's friends from like elementary school. We've known each other forever. We went up probably like 15 years ago or more and saw him. He was doing a show on Broadway, and he invited us to come up and see it. So we came up and we saw the show, and they took us backstage. We got to say hi to some people, and I panicked at the person that I probably wanted to say hello to the most. I completely clammed up because I was starstruck. So it was Patti LuPone. She was as close as meeting you, Emerson, and I just went and froze because she's amazing. But I got to meet Lauren and Nancy. She was very sweet. It was fun. It's a good weekend. I got to see all kinds of stuff when we went out there for that. He's a neat guy. Really nice guy. We have our flat ones today, ladies. They're so cute. I think they're very cute, actually. All right, maybe Evan's just running a hair late. It's got about 1 minute. All righty, let's see. All right, so we're talking about cell division. Cell division, cell division. And so what we're going to do today is this is the end of our first quarter. How many quarters do we have in a year? That's an easy question. How many quarters make a whole? How many fourths make a whole? 4, right? So this is first quarter, and this is our last lesson for first quarter. So what that means is after today, you have till the end of the week, to get your homework in and your test. And once that's all in, then I can give you your progress report for Q1, first quarter, because we've made it through, which means we are 25% through our year already. That's pretty cool, right? Seems like it's going very fast to me. All right, so we will go ahead and start. So today we're talking more about cellular division. We talked about it briefly, and I think your homework assignment touched on it. So we're going to go ahead and talk about cell growth, division, and reproduction. So first, let's talk about some things that limit cell size. Cells grow, right? But they don't grow and grow and grow and grow and grow and grow and never stop growing. There has to be something that limits that growth so they don't grow forever. So the larger a cell grows, the more the demands of its processes increase. When a cell's surface area to volume ratio shrinks to a point where the membrane cannot adequately accommodate for waste removal, the cell must divide. Cell division solves the problem of information and materials exchange overload. So imagine a balloon, okay, and your balloon is made of latex, and you keep putting air in it, and you keep putting air in it. It gets to the point where it's as big as it can be, right? The problem with cells is they have all of those processes going on inside, all those life processes, organelles are doing things, and waste is being produced. And the waste has to be removed from the cell across the membrane. But when it gets to a point where there's more waste than the membrane can manage, the cell's going to divide in order to keep it from basically overloading and exploding or dying. So we have what we call the cell cycle, and this explains how cells grow and divide. So the process of cell growth and division is called cell cycle. And whoops, I didn't mean to hit that button. So if you look here, it's a cycle, that's why it's a circle. And see where it says G1 and G0? These are basically when your cell's resting, and it's part of interphase. You can see here because it's written in green. So all the things that are in green or tealish colors are part of what we call interphase. And that's when your cell is it's resting. It's done, it's growing, but it's resting. It's not doing any of this other dividing processes here. Your dividing processes include your prophase, your metaphase, your anaphase, and your telophase, which we'll talk more about as we go. But these are the actual dividing. Hey, do we turn in live 7? Do you turn in live 7? No, I don't think so. Sorry, I had to think about it for a minute. Yeah. My brain's I've had an overload of stuff this week with stuff with my dad, so my brain's kind of dead at the moment. But all right, whoops, there you go. Oh, and let me give you that. I have your lab 5. Lab 5's over there for you. All right, so there's your lab 5. Okay, process for cell division. So prokaryotic cell division. We're going to focus on prokaryotes. Okay, so what are prokaryotes? Remember, what's it mean to be a prokaryote versus a eukaryote? So your eukaryotes have a membrane-bound nucleus, right? And your prokaryotes do not. So your prokaryotic cells are kind of lesser evolved is a good way to think about them. They're simpler. They don't have a membrane-bound nucleus. Are you a eukaryotic organism or a prokaryotic organism? Do your cells have a membrane around their nucleus? Yes, you're a eukaryotic organism. Okay, prokaryotic cell division. The usual method of prokaryote cell division is termed what? Does anybody know what it's called? Prokaryotes divide? So it's called binary fission. Binary meaning 2, fission meaning to divide in half. So simply means divide in 2, binary fission. The prokaryotic chromosome is a single DNA molecule that first replicates and then attaches each copy to a different part of the cell membrane.
And when the cell begins to pull apart, the replicate and original chromosomes are separated. So it's a very simple process. And here is a picture. All of these little prokaryotic cells are starting to divide. You see how they're like starting to become two? That's what's happening right here. So now we get into eukaryotic cell division, and this is really what we're going to focus on more because this is what happens in our bodies and in pretty much all higher organisms. So eukaryotic cell division occurs in stages. You have interphase, meiotic phase. Mitosis can be divided into several phases. So you have an interphase, this is your resting phase, and then your meiotic phase. Your meiotic phase is when your cell is dividing, okay? And your mitosis is divided into prophase, metaphase, anaphase, telophase with cytokinesis. So cytokinesis is when it actually divides, splits into two. So let's have a look at these a little closer. So interphase. Remember I said in interphase cells are doing what? They're just kind of resting and growing. They're not dividing. There's no active dividing in interphase. So this is which appears to the eye to be the resting stage, primarily resting. Cell just doing what it does, just living its life, right? It's actually a period of diverse activity because it's when the cell is doing what it does. So if the cell is tasked with, if it's part of a tissue that, say it's part of your kidneys, and part of its job is to form tissues that help clean your blood and filter your blood, then it's actively doing those activities. So there's a lot going on, even though it's resting, right? Those interphase activities are indispensable in making the next mitosis possible. Interphase generally lasts at least 12 to 24 hours in mammalian tissue. During this period, the cell is constantly synthesizing RNA, producing protein, and growing in size. So for every half a day to a day, any given cell in your body, somatic cell, body cell, is just going to be sitting there doing what it does, right? Just doing what it does, not dividing, but doing its job. All right. And then what happens is you get to prophase and see what's happening here in prophase. Prophase is where the complex of DNA and proteins contained in the nucleus, known as chromatin, is going to condense. And then the chromatin is going to coil and become increasingly compact, resulting in the formation of visual chromosomes. The sister chromatids are pairs of identical chromosomes. So look right here at this one. Do you see this right here? This is what your DNA normally looks like during interphase inside your cell's nucleus. But by the time you get to prophase, they're going to look like these little X's that you've often seen chromosomes represented like little X's, right? And really what this is it's one here and one here. So you have, this is a chromatid, this is a chromatid, and they come together and they form a chromosome, but they're called sister chromatids. All right. So the sister chromatids are pairs of identical copies of DNA joined at a point called a centromere. That little dot in the middle is called a centromere. And the main events of prophase are your condensation of your chromosomes. So they came together from that little mass, look like a big knot, right? And then they made this little X shape. These movement of these centromeres, so these appear. And then this formation of what we call a meiotic spindle. See these lines going across right here? Those are, see those little white lines? Those are called meiotic spindles. And it's going to form almost like, it's almost like ropes for tug of war. If you can imagine them being like ropes for tug of war. And this is when the nucleoli start to break down. So your nucleus is going to kind of break down. It's not going to be a thing anymore. So then you get to metaphase. Well, look what's happening. You see what's happening in that cell? Did you see that? Watch real closely. Here's what happens with metaphase. Now, all those chromosomes there, they're going to line up in a perfect orderly line. You see that? How they're right here? So metaphase is the third phase of mitosis, the process that separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Long protein filaments called kinetochore and kinetochore microtubules extend from the poles on either end of the cell attached to the kinetochores. So see the little centromere now? Now what we're worrying about is, this is where your tug of war starts. You see how they've all lined up? Like little army soldiers. And you see these little yellow guys on the side? They've produced they've produced these spindles, which are like ropes, right? And the ropes are going to grab those. And then what's going to happen is they're going to get pulled to opposite ends of the cell. So if you watch, that's what's going to happen. So they start to pull them apart like that. And then sooner or later, they get to opposite ends of the cell, just like this. So this is anaphase. This is the fourth phase of mitosis, and it's the process that separates the duplicated genetic material carried in the nucleus of a parent into two identical daughter cells. We don't call them son cells, we call them daughter cells. And the separated chromosomes are pulled by the spindle, there's your tug of war, to opposite poles of the cell. So you see how they were lined up? And now they're being stretched to opposite ends. See that? So they're being pulled. It's like a tug of war. They're being pulled to opposite ends. And then if you notice, you kind of got a peanut shape now, right? Because all of your cell's material is basically dividing out to opposite ends. Everything's kind of dividing itself to opposite ends. So then you have telophase, and here's where it starts to really pinch off. Now you've got like if it's a peanut with a very skinny waist. It's a corseted peanut. So during telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. So now you see we have a nucleus again. Our nucleus has formed. So all those sister chromosomes, chromatids that were pulled apart to opposite poles, now you've formed a new membrane in each of those ends, and the DNA, which is on those chromosomes, remember those hold your DNA, is now at opposite ends. And then what happens is you get cytokinesis, and that's when the your cell membrane is going to pinch off and allow that one cell to now be two completely separate cells. So it's the physical process of cell division, cytokinesis. And in which the cell divides the cytoplasm of the parental cell into two daughter cells. The contractile ring shrinks at the equator of the cell, pinching the plasma membrane inward and forming what's called a cleavage furrow, which then just. So this point is called cytokinesis. We also call it cleavage. Cleavage means to cut in half. So they've been cleaved. They've been cut in half. Now let me ask you, do you think these two cells are genetically identical or genetically different? What do you think? What do you think? Identical? What do you think, Everly? Do you think they're genetically identical or genetically different? Not sure. Emerson, what do you think? You think they're identical? They're identical because remember they started with the same DNA in that one nucleus, right? And then we organized it into chromosomes, and we divided those sister chromatids out, and now they're in their own cells. But this is identical. This is not how we produce gametes, which are sex cells like sperm and egg. This is how we produce identical cells for the purpose of growth, right? All right, so you've got right here, you've got yourself two identical cells. We've just divided, that's all. All right, so here's an Amoeba Sisters version of what we just watched. Watch it again. Do we get a commercial?
JrLR, attorney John Foy and
Go John Foy.
You were just doing your job when an accident took.
Have you ever looked down at your leg or your arm to find some cuts and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it. This amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes, very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells. And in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So, in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Did you know that that's what cancer is? Uncontrolled cell growth? Hello, my mouse just disappeared on me. Go. There we go. Go to the next one. No, no, no. Oh boy. Why is my, hold on. Oh no, sorry.
Performance, jerk,
Trying to make this go here and it doesn't.
ability, afford it. Only one place. That's
Hold
it all.
on.
Whatever you do, do.
Yeah, we just did that. Stop. My mouse disappeared and now it won't move. Hold on. Let me try and get it to, hold on. Oh, stop. It's gonna make me crazy. Stop, stop, stop. Why won't it go? Hold on. Oh my. Oh, there we go. Hold on. All right, here we are. Back to where we need to be. I don't know why my mouse just died for a minute and then it came back to life. Okay, so she said PMAT. You have to remember that the I comes first because that's for your interface. So some things people have come up with in the past is intelligent people make all things clear. So that's interface, prophase, metaphase, anaphase, telophase, cytokinesis. Improper poopers must always think clean. I love that one. That's a good one. If you like gross stuff, you might remember that one. Improper poopers must always think clean. Then we got, I punched my annoying tennis coach. Some people use that one. That one's not very kind, but you might remember it. I play Minecraft at the computer. That's a good one, right? So they were saying that cancer is uncontrolled cell growth. This is a shorter one, but it shows you how that happens.
Take 5 minutes to get the picture. More than 60,000 billion cells make up the human body. They are the units that form the tissues which make up our organs, like the liver, heart, and lungs. When the body needs it, our cells split into 2 and replace those cells that are defective or have come to the end of their lifespan. This makes it possible for our tissues to preserve their shape and their respective functions with the passage of time. Every cell is therefore programmed to multiply and die. This ordered but complex program is controlled by the center of the cell, the nucleus, which contains chromosomes containing many genes made up of DNA. Sometimes some of these genes undergo a change. The nucleus then sends out abnormal orders and the cell goes wrong. It multiplies uncontrollably and takes on a life of its own. Each new cell produced contains the same defect. The cells proliferate chaotically and form a tumor. This process may be short, but is often long. 10 to 30 years may separate the birth of a first abnormal cell from the appearance of a tumor of about 1 cubic centimeter. The tumor forms many blood vessels in order to survive. These will supply the tumor with oxygen and nutriments, allowing it to live and grow. This is what we call the phenomenon of angiogenesis. The tumor, however, only really becomes dangerous when the cancerous cells begin to invade through the vessels into the adjacent areas and spread to the surrounding organs. These cells can then invade other parts of the body, multiply and produce new tumors. Metastasis is the term used for this spreading process. But why does a cell become cancerous? There are many factors, and they can exist to different degrees. Factors have been identified, such as hereditary genetic anomalies, exposure to some viruses, HIV, hepatitis B, C, and D, papillomavirus, Epstein-Barr virus, exposure to toxic agents, chemical products, radiation, sun, unhealthy behaviors, such as the consumption of alcohol and tobacco, or a diet too rich in fat and low in fruit and vegetables. Today, over 25 million people in the world live with cancer. 7 million die from it each year. The disease represents the leading cause of mortality for those under 65. Lung cancer is the most widespread, followed by breast cancer, colorectal, stomach, and prostate cancer. The means to combat cancer do, however, exist. These often complementary treatments are used alone or in combination according to the type of cancer and its status. The goal of the treatments is to make it possible to eliminate the tumor and cure the patient in early stage cancer, or like a chronic disease, to control its development. There are mainly 3 kinds of treatment. Surgery consists of removing the tumor in part or in its entirety. Radiotherapy exposes the tumor to radioactive rays, which prevent the diseased cells from multiplying and destroys them. Chemotherapy consists of administering systemic medicinal substances to destroy the cancerous cells or prevent them from spreading. Chemotherapy and radiotherapy act on cancerous cells, but also on healthy cells, which explains their side effects. But today, better knowledge of the characteristics of cancerous cells makes it possible to develop therapies that target the cancer development mechanisms more specifically. Sanofi Aventis R&D is already present in all these areas to fight against cancer on all fronts.
So the 3 big things I want you to remember about cancer is that it's either genetic or environmental. Okay, so it can be something you're just genetically predisposed to, and there's nothing you can do about that. But it can be environmental, like chemicals, things you consume, like alcohol, cigarette smoke, all that kind of business, sunlight, all those things, right? That that those things, whatever those things are, either your genetic predisposition or whatever you come in contact with in the environment, cause damage to your cell's DNA, which cause it to replicate uncontrollably, uncontrolled growth, uncontrolled replication. Okay, so it's either environmental, and it's either environmental or genetic. That's number 1. Number 2, it's uncontrolled cell growth. And number 3, skin cancer. And the reason I'm telling you this is because you right now can help prevent yourself from getting skin cancer. What do you do to prevent skin cancer? What do you do? You put sunscreen on. But here's the thing that matters. Remember when they said that sometimes it takes 30 years before you see the damage? Like the damage begins, and it can take 10 to 30 years sometimes before you're aware of the damage. Skin cancer is one of those that you develop as an adult as a result of sunburns you had as a child. You can get sunburn as an adult. And it's not, it's gonna take so much time down the road that it's not gonna be as big an impact as it is when you're a child. So the sunburns you get in childhood are the ones that can cause you cancer as an adult. So people think, oh, it's just a sunburn. We all had sunburns. Yeah, try to avoid them because that could cause skin cancer down the line. So that's something you can do today. So whenever you go outside in the sun and you're like, oh, it's just sunny, whatever, I'll be fine. Put on some sunblock or stay in the shade or wear longer, light colored long sleeve clothing or whatever. I know multiple people, like my father-in-law had a big chunk of his nose done, and I have a dear friend right now who just had 2 weeks ago. She had on her nose is a very common place for skin cancer. She had a piece here removed, and what they do is they numb you up and they start slicing away at it, cutting out the cancer, cutting up pieces of your skin, and then they look at it under a microscope and they keep looking. And if they still see cancer, they cut deeper. And they let you go wait in the lobby while they look under the microscope. And if they still see cancer, they call you back, they cut some more. They had to cut all the way through her nose into her nasal cavity, like down into her nostril. She had a hole all the way through. So then they had to take a graft from over here by her collarbone and put that in. And the graft isn't taking well. So she's had bandages on her face for 2.5 weeks now. And it's really, really, really awful. So that's one that you can try to avoid, like seriously. All right, fate mapping. So cell differentiation, I want to tell you what fate mapping is. So we're going to talk here about how when your cells start to divide in an embryo. So this is how you grow from being a single fertilized cell, which everyone was at one point, to 2 cells, to 8, to 4 cells, to 8 cells. Everybody looked like this at one point in your life. We all did, right? And then that's how you start out, right? You start out as an egg and a sperm that came together, and there you are. And you just continue to grow. So here's what fate mapping is, and it's really interesting. So you see all these little cells here? They are programmed to develop into a certain type of tissue. And it's different for every one of those cells. So fate mapping is something you can do in a lab where you can take a bioadhesive dye that only adheres to living tissue. And you can say, oh, no. Okay. You can say, for example, at that 8 cell stage, you can go in with a microscope with an embryo, usually like a frog embryo. That's what I used when I did this, was a frog embryo, because they develop externally, right? You can watch tadpoles grow in the water. They don't develop like inside of a mother. So you can go in and you can tap a teeny tiny little microscopic drop of that dye onto one of those 8 cells and then see what it turns into. One of them will become a nervous system. Another one will become a digestive system. Another one will become skin. And it's so cool to watch. You can watch it every day. You can go look under your microscope once a day and watch that embryo grow. And whatever part of its body is, like the dye we used is called Nile blue. It was bright blue, like Evan's shirt, bright blue. And you could look at that little frog embryo every day, and it would be, parts of it would be blue, depending on what that cell was fated to become. That's called fate mapping. It's really fun. So this is interesting too. So these are human embryos under a microscope.
Opioid addiction gets between you.
Oh yes, opioid addiction is very bad. Don't do drugs. We know that.
The journey of human embryos, as seen under a time-lapse microscope.
So remember, even you looked like this at one point, right?
A human egg, as shown here, is fertilized by either conventional IVF or intracytoplasmic sperm injection.
So in that video there, they're injecting
This
one
egg
sperm
is
into an egg in a lab.
16 hours post-fertilization. The egg highlights signs of fertilization as the male and female DNA come together. At around 24 hours post-fertilization, the fertilized zygote undergoes the first division to form into cell embryo. Embryo continues to divide, and 2 days following fertilization, embryo reaches the 4 cell stage.
So it's going to keep dividing like that. And that's so that we all look like that at one point. All right, so to check these answers real quick, number one, circle the letter of each sentence that is true about cell growth. What do we have for number one? What do you got, Everly? B, B, and A. A2. So for living things to grow, their cells must grow and divide, and after interphase is over, cells grow to their full size. All right, number two, read each word in the box and in each sentence below, fill in the correct words. The regular series of growth and division in a cell is called, Everly? The cell cycle. The first stage of the cell cycle is called, Emerson? Interphase. When a cell makes an exact copy of its DNA, it's called, Emerson? Replication. In the following sentence, true or false? During the first part of interphase, a cell grows to its full size. Everly? True. All right, and then over here, look at these two cells and circle the letter of the cell that has just finished mitosis. Which one has just finished mitosis? What you think? B? Good job. All right, circle the letter of the sentence that's true about mitosis. What do we have for number 5? Anybody have it? So it's going to be A and B. The nucleus of the cell divides into 2 nuclei. 1 copy of DNA is given to each new nuclei. During cytokinesis, the what divides? What you got, Everly? Cytoplasm, right? After cytokinesis, each what has the same number of chromosomes? What you got? Each daughter cell. Very good. All right. Picture shows the structure of DNA. Circle the nitrogen, and so they're circled here. You can kind of see them. 1, 2, 3, 4, 5. These are your nitrogenous bases, or your nitrogen bases. Which nitrogen base pairs with adenine? This is the answer's already up. It's thymine. Do you have it? Yep. Okay. DNA unwinds and replicates. Nitrogen bases floating in the nucleus pair with the unpaired nitrogen bases on each half of the separated DNA. And when a new base is attached, 2 new DNAs are formed. Okay, so real quick, here we go. You have a test coming up. These are the things you need to know. Stimulus and response. Remember, all living things respond to stimuli. That's one of your definitions for living things. Living things grow, reproduce, respond to stimuli. Active and passive transport. Active transport requires what? Across the membrane. Active transport is going to require some level of energy across the membrane, right? Right. Steps of cell division. What are our steps of cell division? Somebody tell me who just did it. Starts with I. Remember, what was it? I punched my annoying tennis coach. Improper poopers must always think clean. What's I? Interphase. Very good. What's next? P, which is prophase. What's M? Metaphase. Okay, then what? Anaphase. Telophase. What's the last one? Cytokinesis. Let's do them again. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis, right? So, which one do you like? I punched my annoying tongue. Didn't you come up with one for homework? What'd you come up with? Anybody remember? I remember. So,
I like the one.
you like them? What was it? I play Minecraft on my computer. I play Minecraft on my computer. So I, interphase, P, prophase, Minecraft is metaphase, on is nothing, my is, that's right, anaphase, at my computer. That's right, that makes sense. Anaphase, metaphase, telophase. So I play Minecraft at my, that makes sense. All right. ATP and cellular energy. ATP is your source of energy, remember? It's part of cellular respiration, Krebs cycle. We looked at all that last week. That stuff's a little dry. But remember that ATP is your source of cellular energy. That when you go from ADP to ATP to ADP, that's when you're building up energy in your cell. And remember that there is that it's all has to do with electrical potential in the end. It comes down to where the electrons are. Do you remember? All has to do with electrical energy. So photosynthesis versus respiration. Why do you think I said versus in there? Not just and. What about those two things? What's significant about comparing or contrasting photosynthesis and respiration? Do we remember? We don't remember. Nobody remembers. Do you remember that there are chemically practically opposite, right? That in photosynthesis, you take in water, right? When you take in carbon dioxide, water, and what do you produce? Sugar and oxygen. And then with cellular respiration, you take in oxygen and sugar and you produce water and carbon dioxide. Okay, know that. Prokaryotes versus eukaryotes. We just said this earlier. What's a prokaryote and what's a eukaryote? Somebody tell me. Prokaryote. What's a eukaryote have that a prokaryote does not have? A membrane-bound nucleus. Remember, all of the DNA and everything is inside of a membrane in the nucleus. Which is more highly evolved, prokaryote or eukaryote? Eukaryote. Good, Emerson. Eukaryotes are going to be more highly evolved because they're more complex. Plant cells versus animal cells. What's your big difference between plant cells and animal cells? They're kind of two, but yeah. A cell wall. Plant cells have a cell wall. Plant cells also contain chloroplasts, right? And they also have that big, large contractile vacuole. Remember that big vacuole. What's the purpose of that big contractile vacuole? Do you remember? Kind of it holds the water in the plant, keeps the plant's form and shape, sturdiness, right? It's called turgor pressure. Organelles and their functions. Know about your different organelles. Remember back to when we talked about how they were kind of like workers in an office? Yeah. Know that. Smooth endoplasmic reticulum, rough endoplasmic reticulum, vacuoles, Golgi apparatus, chloroplasts. Know what these things do. Organization from cells to organism. How do you get from cells to organism? Cells join together and form what? Tissues. Tissues join together and form organs. Organs join together and form organ systems, and organ systems form an organism, right? Remember that. Taxonomic hierarchy. This is the one where we talked about dear King Philip came over for good soup. Remember that? So it's domain, kingdom, phylum, class, order, family, genus, species. Domain, kingdom, phylum, class, order, family, genus, species. Know that. That's important to know, okay? The reason that's important to know and not just some sort of like rote memorization is that while we're in this class and we're working with different organisms, like today you're working with your first live organism in class, besides just observing them. But it's important to know how organisms are related to one another. So that's why taxonomic hierarchy matters. Binomial nomenclature. Remember that is genus plus species. You say the genus, then you say the species. And that's how you name things. And then classification in general. How would you classify something? Producers, consumers, decomposers, scavengers, heterotrophs, autotrophs. Remember that autotrophs are producers, heterotrophs are consumers or decomposers or scavengers, correct? Everybody remembers that? If I showed you a picture of a chicken, would you say it's a heterotroph or an autotroph? Chicken. Does it make its own food? So it's a heterotroph. Would you say it's a consumer, decomposer, or producer? Producer. Chicken? Say it's a consumer. It eats food, right? Okay, so keep that in mind. DNA base pairs. Okay. Remember that this was very briefly discussed in this one, but DNA base pairs. Adenine goes with thymine, guanine goes with cytosine. So A with T, C with G. A with T, C with G, G with C, T with A, right? They always go. Yeah, we did that in chemistry. So remember, C and G go together, A and T go together. Keep that in mind, okay? All right, we're good. You can get your lab out.